Okay, now this will be the lecture 3 for the class Computational Biology. And the date of the class will be February 17. So in the previous two lectures we said a few things about next generation sequencing, and we had a very short introduction to Galaxy. Now we will continue with Galaxy, and we will proceed to the first steps of the processing of the NGS data. So the first thing that we had seen last time was how to load data into Galaxy. To remind you what we did, we went here to the upload button, and then we copy paste some links here, but first we went to this paste fetch data. We clicked on that and we copied paste the URLs of the data here. Now the question is where to find these URLs. The usual answer is we should go to the ENA nucleotide archive. Okay, here. Then we search for a dataset. The dataset we were using was this flour, yeast, genuine project. Okay, it's this study here. This project comes with some data. Here they are. And what we had done is that we had taken some URLs. I will just take manual here, but in the last class we had seen how, or I can show it, how we can automatize a little bit this process. So what we can do is that we can download this information here in tab separated values. We can save it. It's actually this file. And then we can go to Excel. I'm using Google Sheets. And do the following. In principle, this process is better to do it in Linux, but since students are not familiar with the command line, I will just use the Excel. We will have the same result. So file, open, upload, browse, and then we go to home. I'm just looking for the place that I saved table. Here probably I need all files. It's not this one. Where's that? And this should be this one or this one. They are exactly the same. Probably I should change the suffix of the file. Okay, let's do it again. Okay, it seems that Google Sheets looking for an XLS file. It's okay, no problem. We'll open it, copy paste the contents. It's easy to paste it here. Okay, and then we are interested on this column here, the H column, which is the URLs. I copy it, I paste it here to have it clean, and now we notice that actually here there are two links separated by semicolon. So what we will do is that we will select these cells that contain this information, and then I will go to data, and then split text to columns. In Microsoft Excel this is a bit different. Again, you go to data, then you see a button somewhere here writing split text to columns or something similar. Okay, and then the separator is a semicolon. And now you see that actually there are two columns, this one with the first link and this one with the second link. So these are the two paired files because we have paired and experiment. Just cut this and paste it here. And there is one more thing to do. So the link does not contain this http colon / or ftp colon / actually here the link should look like that. So what I will do in order to change this is that I will replace with Control H the ftp.sra with ftp sra. So then I will introduce this ftp colon / in the beginning. Okay, now these are all links. So now I can copy it. Copy all the links, go to Galaxy and paste them here. Since I have done it already, I will just demonstrate it for a few. What I can do probably is I will delete all this and I will just upload the pairs for a couple of files. So I go again to Excel. Just take the first two and I will delete all the rest. Delete and also will keep the first two from the reverse read. And I will delete all the rest. And I will delete these columns, these rows, delete, where is it? Delete row. So I will have these 4 files, 2 forward, 2 reverse, 40, 40, 41, 41, it's fine. Just to demonstrate how I will upload them here. Okay, I'm regular tab. Can go in the collection, but I can later on save them as a collection, so there is no need for that. And go and close. So here I see that these files will be uploaded. It will probably take some time, but they will be uploaded eventually. Don't pay attention to what is running here, it's one of the next steps for the analysis. We will wait here and until this file will be uploaded. Okay, now the uploading of these 4 fastq files has been finished. We can even view them just pressing this eye here. See that these are short reads, Illumina short reads. And now what we can do is we can select these 4 files and make a collection of perdent files. Let's see how to do that. So we select these 4 files. We can even use the shift. I have pressed the shift now, and then I click this last file here, so all of them are selected. And then I have this option here that asks me what I want to do with these 4 files that I have selected. Click here and I click on build list of dataset pairs. So it automatically recognizes this 1 and 2. I just type a name here, test yeast. And this is the file. It's a list with 2 pairs. This is 1, the forward, the reverse. The benefit of this process is that we can apply things on all the files of the list. Okay, so the first thing that we will do is to apply some quality checks and filtering, but we will do that in the next video.